Hello students. In this video, we are discussing a question from the CSIR syllabus, Unit 11, that is Advances in Biotechnology. So today's question is, live vaccines are generally used to protect against which one of the following viruses? And our options are option 1, hepatitis A, option 2, hepatitis B, option 3, rabies, and option 4, that is smallpox. Now, many of you might already know the answer, but before going into the answer, let's talk a little bit about vaccines because it is kind of related to the answer. So, the word vaccine was first used by Edward Jenner. He was the scientist who discovered the vaccines for smallpox. And what he observed was that uh, milkmaids who worked with cows, they used to get a milder version called cowpox, which has somewhat similar symptoms to smallpox, but it wasn't as dangerous. And the milkmaids never used to get the smallpox. So what he did was he took pus from one of the milkmaids as an inoculum and inoculated an 8-year-old boy. So that was the first modern use of vaccine. And the word vaccine comes from the Latin vacca, which means cow. So now we'll dive a bit into the type of the vaccines. So at larger level, there are two types. One is where we use whole organisms, whether be it bacteria or virus, as the vaccine, and another is where we use only components of the microorganism instead of the whole organism. Now, looking into whole organisms, there are mainly three types. So one is attenuated, that is weakened bacteria or virus, but there are they are still alive. But either they have been weakened or passaged through different hosts so as to lose the virulence. And second is outright killed organisms which have been killed by either heat or chemical treatment. And third type is recombinant vectors, where uh, by genetic engineering, the antigen is expressed into a similar type of bacteria or virus, but one that is non-pathogenic for us. Now, moving on to component vaccines. So, first type of the component vaccines is toxoids. So, some of the bacteria they produce the exotoxins, and in the toxoids, these toxins are purified and they are heat inactivated to produce toxoids, which can be directly used as vaccines. Now, the second type is protein vaccines, where surface proteins of the bacteria or virus can be used as a vaccine. And one example is one of the hepatitis B vaccines. Now, third is capsular polysaccharides. So many bacteria contain capsular polysaccharides, which are antigenic, and they can be used as vaccines. The example of this will be the vaccine for pneumonia, which contains 23 different antigenic capsular polysaccharides. Then we have synthetic peptides. These type of vaccines are mainly used against cancer cells. You can consider them as a kind of personalized therapy where we can look into novel peptides which are present in yeah, individual cancer types for different persons and use them as vaccines. Then, in the end, we have mRNA vaccines and DNA vaccines. So, in both these cases, we directly provide the code for synthesizing the proteins, and the host body prepares the antigen from these, and it induces the immune response. And the examples of this will be certain vaccines against certain viruses which caused global lockdowns a few years back. So now we'll look at the viruses which are our options in this question. So first, a little details about hepatitis A. So its genetic material is RNA. Strand polarity is a positive strand, and its nucleocapsid symmetry is cubic, and it doesn't have an envelope around the capsid, so it is called naked versus the envelope. And the vaccine for this is a killed virus. So, in regards to our question, this is out of the option. We are looking for live vaccines. And then we have hepatitis B. Its genetic material is double-stranded DNA. Nucleocapsid symmetry is again cubic. Now, these viruses have an envelope made of plasma membrane of the host cell, and their vaccine is again killed viruses. Then we have rabies. So its genetic material is RNA, and the strand polarity is negative. Its nucleocapsid symmetry is helical. It also is an enveloped virus, means it will have a layer of host membrane around the capsid, and the vaccine for this is in the form of killed virus. And at the last, we have smallpox. Its genetic material is DNA, double-stranded DNA. The nucleocapsid symmetry is complex. This is also an enveloped virus, and its vaccine is a live weakened virus. So, this is the summary of all the four viruses. If you want, you can take a screenshot of this for future revisions. And again, coming back to our question, the correct answer will be option 4, that is smallpox virus. Now, if you want me to make video about any of the vaccine types that you would like to know more detail about, let me know in the comments. If you like the video, press the like button, share it with your friends. Also, if you are visiting this channel for the first time, do subscribe. I try to post such educational videos daily. Thank you for watching.